Alright, so we're going to continue from last time. We are in chapter 29 now, which is transcription. Specifically, chapter 29 talks about prokaryotic transcription mostly. At the very end of 29, they begin to introduce to you eukaryotic transcription. Before we get into the mechanism of transcription, we need to understand this one concept here, which is the difference between sense strand versus antisense strand, and why we call one thing sense strand and one thing the antisense strand. So, I've drawn an example DNA sequence here, double-stranded DNA here, and I've labeled which end is 5' to 3' and which one is 3' to 5'. If I want to transcribe this DNA sequence, what's going to happen is there's an enzyme called Rna Polymerase. Okay, there's an enzyme called Rna Polymerase. Let me zoom in some. That's going to bind here and begin reading this strand in that direction. Reads template in that direction. It's going to move along from left to right on this picture here. You guys remember with DNA polymerases, any polymerase, if their job is to make a nucleotide sequence, DNA polymerases were synthesizing brand new DNA. RNA polymerase is going to synthesize RNA, and in this example we'll say that it's making mRNA. Those polymerases always read a template strand in the 3' to 5' direction, because when they generate the brand new strand of a nucleotide polymer, it has to make it in a 5' to 3' direction. So it reads the template in that direction, but makes new stuff in this direction, 5' to 3'. So, for RNA polymerase, it's going to read this T here on the template, and what is it going to put on the RNA? What is the complementary base to T? It is A. What's the complementary base to A, though, if we're talking about mRNA? It is uracil. Okay, be very careful with that. It is uracil. And, I'll just finish the rest. G, C, A, U, C, G. This is our mRNA that was generated in a 5' to 3' direction because it read this template 3' to 5'. At a later date, for whatever reason, a different RNA polymerase can go up here and read this one as a template strand, just because this strand is used as a template in this example. Just because the bottom strand is used as a template in this example does not mean that the top strand will be ignored always. Okay? Certain genes are located on one strand versus the other. In this example, I'm going to just, label this as the positive strand. And this one down here is the negative strand. This is kind of the notation they use in genetics and molecular biology to distinguish which strand of DNA you are looking at your sequences for. Which strand of DNA will be the template strand that an RNA polymerase will bind to and read. Some genes are on the plus strand of DNA, meaning some genes, the RNA polymerase will have to bind to this strand to read as a template. And other genes are going to be on the minus strand of DNA, so RNA polymerase will have to bind that one instead. Don't worry about memorizing which direction is plus or which direction is minus. Just note that that's the notation that is used to distinguish one strand from the other. In this example, this RNA polymerase is binding to the minus strand of DNA to read this template to make brand new mRNA. Well, I've used the phrase template to describe this strand down here, this DNA strand here. You should memorize that would mean the antisense strand. Template means antisense strand. Okay? And whenever we use the phrases antisense or sense strand, we are talking about transcription, not DNA replication. We are talking about transcription. That's a common mistake I see all the time, is people start using these phrases when they're talking about DNA replication. This is only for transcription. That means this strand up here, the non-template strand, is your sense strand. And here's why it is called the sense strand. Compare this sequence with the mRNA you just made, and you will notice something very quickly. They're the exact same sequence in the same direction, except for one slight modification, and that is your mRNA has a bunch of uracils in the places that the sense strand had thymines in it. All right? So, by definition, the sense strand of DNA will look similar to the mRNA, just with that slight modification. The antisense strand is complementary to the mRNA, meaning that you have to read each base to figure out what its complementary base is. So the antisense strand is complementary, while the sense strand is equivalent to the mRNA. All right? Let me think if there's any other vocab terms to give here. So if you see these words used in any questions, note that this diagram kind of breaks down what each one means. All right, let me flip back to the PowerPoint and make sure I didn't forget anything regarding this. Convention, blah, blah, blah. This is an old convention and will not be used on the exam. I will, because it is very confusing, because what happens if they draw it the other way around? Do not memorize that always the 5' to 3' as drawn is your sense strand and the 3' to 5' is your antisense strand. Do not memorize that. Look at which one is the template strand that RNA polymerase is bound to. The template strand is your antisense strand, and the non-template strand is the sense strand. The reason why notes will use these directions and these names is because most of the time they draw your DNA strands like so, like drawn in this diagram. So for this particular diagram, notice that the 5' end is in the top left, which is what we drew earlier. Well, let's see. The 5' to 3' end is your sense strand. That's what we drew earlier. So for this diagram, that's correct. And the 3' to 5' strand end, which is the bottom one on this diagram, the one that RNA polymerase is on, that is your antisense strand. That's what we happen to draw up there. I will stress that this is not always true, because what if I decided to flip it around? Okay? A lot of people will think, like, for example, maybe the RNA polymerase is going to bind here and go that way. Okay? Using the rules up there is not going to help you, because that would mean, if you use the rules up there, that this up here would be your antisense strand. Whoops. Right? Because it said 3' to 5' must be your antisense strand. Okay? It is very confusing, so I would go with just look at where the template strand is. That is your antisense strand. All right? So it's an old notation. All right, let me make sure I didn't forget anything else. That's it for that. So that's the start of how to break down sense strand, antisense strand, what's being transcribed, what the mRNA looks like. Now we get into the fun stuff, and that is the actual mechanism of transcription and the regulation of transcription. Okay, so first and foremost, remember how I said that a lot of these mechanisms and symbols are going to look similar to each other? Look at that. We have Greek letters again. This is the third time. If you go through the notes, this is the third time Greek letters are showing up. The first time you saw it was in prokaryotic DNA polymerase 3. The second time you saw it was in eukaryotic DNA polymerase, all those eukaryotic DNA polymerases. And now you see it in prokaryotic transcription. I stress again, separate your notes into different sections, into different sheets of paper, and compare them to each other, because Although it looks like, scientists are lazy, and they are, they use the same names to describe completely different things. When you look at the definitions of these things, you can associate specifically one Greek letter in one context with something. So I'm going to try to break that down for you here. In prokaryotes, there is a single enzyme that transcribes everything. And that is just called RNA polymerase. No Roman numerals and no Greek letters in the name. It's just RNA polymerase. So whenever you see written RNA polymerase, that is just prokaryotic transcription. I can't tell you how many times that when someone sees RNA polymerase Roman numeral 2, they think, well, DNA replication had Roman numerals with DNA polymerase. That must mean it's a prokaryote. No, and I don't know why they decide to label these things so confusingly. Prokaryotic transcription is just RNA polymerase, no extra numbers or Greek letters after it. RNA polymerase is prokaryotic transcription. And you'll see what I mean when we get to eukaryotic transcription, how it gets more complicated. The Greek letters come in because the notes describe that this one enzyme is made of a bunch of subunits. So, similar to DNA polymerase, right? DNA polymerase 3, you learned that one was made of a bunch of subunits with Greek letters, right? There was an alpha subunit in DNA polymerase 3. There was a beta sliding clamp. There was epsilon and theta there. Different Greek letters. Well, some of them are the same Greek letter, but they mean different things here. That's why you have to look at the context of the question or the answer choice that you are reading. These Greek letters are subunits for RNA polymerase in prokaryotes. Here's what they do. The alpha subunit, think of it as it's the structural component of RNA polymerase. There's a nice definition for it, but it is the structural component. It brings everything together for RNA polymerase, the alpha subunit. Okay, that's this green one right here. Beta and beta prime each bind to different things. Well, what's RNA polymerase's job? RNA polymerase's job is to read a DNA template to make mRNA. So 2 binding events have to happen. One, some part of this enzyme needs to bind to the DNA template. In this case, it's the beta prime subunit of this thing. And another part of this enzyme, the beta subunit, needs to bind the nucleotide triphosphates, as in the mRNA nucleotides that have to be brought in and put together to make mRNA. So beta prime, its job is to bind to the DNA template. This one brings in nucleotides so that the enzyme can start putting them together to make mRNA. So beta binds the brand new RNA nucleotides. The sigma subunit is rather interesting because it is technically separate from everything else. And we'll see some diagrams that better illustrate this. This is just text definitions right now. The sigma subunit technically comes in later. And if you read it here, it says it recognizes a promoter sequence. Well, what's a promoter sequence? What does that have to do anything with transcription? I think it actually is probably the most important thing with transcription in prokaryotes or eukaryotes is promoters. So let me skip to where they talk about promoters first, and then I will go back. All right. Here's a diagram. For those who aren't familiar with these types of diagrams, this is a diagram illustrating transcription at a particular gene. I'm going to break down what it's showing you here. This purple stuff is a gene on DNA that needs to be transcribed. Okay? Meaning RNA polymerase will have to somehow read this template. I guess it would be the bottom strand here in this case. It has to read this template to make brand new mRNA. In order to do that, it needs to know where this site even is on the genome. How does it know where to find this site? Every gene that is transcribed has a site called a promoter region directly what is called upstream to this gene, meaning right before this gene exists a series of DNA sequences called promoter sequences that are kind of like the landing pad for RNA polymerase to bind to. Promoter sequences contain special regulatory patterns that RNA polymerase is looking for to figure out where to land and where to start transcribing. When it finds a particular site with a particular promoter sequence, it will bind to there and then transcribe anything that is after that, okay? So you see these phrases upstream and downstream. Whenever you see the phrase upstream, it means that it is before the gene, relatively speaking. and downstream meaning it is down the gene, after the transcription start site. So it's all relative stuff. We'll get into more examples of upstream, downstream when we get to eukaryotes. But all promoter regions are directly upstream of the gene. Let's see, anything else? Oh yeah, every gene you have to finish transcribing as well, so eventually there's going to be a site way downstream, meaning the very end of the gene, that is called the transcription termination site. This is a site at which RNA polymerase will stop transcribing this gene and you will have created your finished RNA transcript. That involves a mechanism that we'll go over later today. All right, so promoters are regulatory sequences that are like landing sites for RNA polymerase. So before we get to the steps about that, let me find this slide here. So this is a zoomed in site of several different promoter sequences, okay? So imagine to the right over here at these green boxes. These green boxes represent the exact base that transcription will start at for whatever gene that they're talking about. Doesn't matter what gene it is. Each of these green boxes represent the transcription start site. So everything to the left of it is upstream of that transcription start site. You'll notice that whenever they talk about upstream things, they use negative numbers to describe that because that means it is before the transcription start site, which is indicated by a +1, which is the very first base that will be transcribed. The minus numbers indicate how far upstream it is. So there's a region in the promoter called a Pribnow box that is found exactly, or approximately I should say, 10 base pairs upstream of the transcription start site. And there's another unnamed one called the -35 region in prokaryotes. So why these specific regions, why are they important? Well, it turns out in promoters, there are certain parts of the promoter that have a specific pattern that is repeated over and over again everywhere throughout the genome. That is called a consensus sequence. A consensus sequence is a specific pattern that is seen over and over again, more than it is expected to be. In other words, if you put a random bunch of bases together, you would not have a consensus sequence. These have a functional purpose. A consensus sequence is a functional purpose because it's a pattern that shows up more often than you would expect from random base distribution. Well, what would the Pribnow box consensus sequence look like? Well, the way they figure this out is they look at all of these promoter sequences put together and they average out what bases are found more often than not at each of these positions. This sequence right here in yellow at the bottom here is the consensus sequence for the Pribnow box. You don't have to memorize the consensus sequences in this class. You might have to in a genetics class, but for this class you don't have to memorize them. You just have to know what a consensus sequence is. It is a repeated pattern found in a promoter region that an RNA polymerase is looking for. An RNA polymerase is looking for a consensus sequence in the promoter because it uses that to figure out where to bind to. It uses that to figure out where to bind to and begin transcription. They are looking for consensus sequences. Okay? Now, the cool thing with transcription is that the individual sequence does not have to look exactly like the consensus sequence. So you might notice some of them look exactly like it, like for this gene, which is blurry because of the quality of the picture. But for this gene, it has a sequence of TATAAT, which is exactly the same as the consensus sequence. But let's contrast that with this second gene from the top here, whose Pribnow box is TGTCAT. That looks quite different from the consensus sequence of the Pribnow box. It is this slight variation leads to an interesting result here. If the particular Pribnow box looks exactly the same as the consensus sequence for it, that means that gene will be transcribed more often. Why? Well, if RNA polymerase is looking for this exact sequence, and it finds a particular gene whose promoter sequence looks exactly like it, it's going to bind there more often. So therefore, it's going to transcribe that gene more often. When a particular promoter sequence looks more similar to the consensus sequence, it means it is a strong promoter. So if you ever see the phrase strong promoter or weak promoter, Strong promoter means you look at an individual promoter sequence here, and the closer it looks to the consensus sequence, the stronger it is. It means the gene is transcribed more often. Contrast that again with a particular promoter sequence that looks quite dissimilar to the consensus sequence. That represents a weak promoter and one that is not going to be transcribed very often. All right? This subtle little base differences lead to all those differences in gene expression, to where you have some genes that are made abundantly and some that are not. Okay, this is one of that long list of things that affect gene expression. This is one of them, is purely what does the promoter sequence look like. So you can get this in multiple different places. You can have a consensus sequence in the Pribnow box. You can have a consensus sequence in the -35 region. You will learn in eukaryotes there's way more than this. In eukaryotes, there's hundreds of thousands of these. In prokaryotes, for this class, these are the two named consensus sequences that you have to know. So if you see, quote, -35 region, that is a consensus sequence in prokaryotic promoters. And if you see, quote, Pribnow box, or the -10 region, that is another consensus sequence in the promoter region of prokaryotes. So this is all prokaryotic stuff. Okay. Both are necessary for RNA polymerase to bind to this region of the genome in order to transcribe whatever gene is after it. I believe you may even need to know what the functional difference is between these two. Let me see if that is necessary for you. Yep, there we go. So I will get to that right now. All right, so what is the functional difference between these two? You need both, but what is the functional difference? The -35 region, functionally, is where RNA polymerase is going to actually bind to via its sigma subunit. The sigma subunit we saw earlier, its main job is to bind to the promoter, and it's specifically looking for the -35 region in the promoter. The Pribnow box, its function is after the RNA polymerase has bound to this -35 region, the Pribnow box is going to interact with the RNA polymerase to begin unwinding DNA. Because remember, if you want to transcribe, you only want to transcribe one of the strands of DNA, so you have to actually get to it. You need to in order to get to the one strand of DNA, you need to open it up. And so it's specifically, DNA is going to open up at the Pribnow box. And, if you want to look at, hydrogen bonding here, you might notice that the Pribnow box is made of a bunch of T's and A's, which I asked this a million times. How many hydrogen bonds are in T's and A's? 2. Contrast that with if this had a bunch of G's and C's, which is 3 hydrogen bonds. That is a lot harder to open up. So evolutionarily speaking, it makes sense that the spot at which you want to open up DNA probably has a lot of A's and T's in it because it's easier to break apart. All right, so those are the functional differences between those two consensus sequences in the promoter. You need both in order to transcribe a particular gene. And again, I will go back up here to remind you guys of the subunits. The sigma subunit is the one that actually binds to the promoter, and then DNA is going to open up so that you can begin transcription. So, now we get into the actual steps of transcription. All right? And there's 3 steps with kind of like a pre-step, like a setup step up here. The binding of RNA polymerase to hollow enzyme at promoter sites. That's not considered as technically one of the steps of transcription. So if you're asked, what are the steps of transcription? It is initiation, elongation, and termination. But in order to understand all this, you have to know that there's stuff going on directly before these things. So what goes on? Let's look at a nice picture here. Okay. Kind of already know what the very first step is going to be. Right? We know that RNA polymerase has a sigma subunit, and it's going around looking for a particular promoter region. It's not illustrated here, but it's going to look for that -35 region consensus sequence, and if it finds it, it's going to bind to it. So notice that RNA polymerase at the sigma subunit binds the promoter region here. In the act of binding to DNA, you form what is called a closed promoter complex. Makes sense, right? Nothing's opened up yet. You initially make a closed promoter complex. So what happens in step 3? Well, you need to open up the DNA. So that happens at the Pribnow box spot. That's what we talked about earlier because it has a bunch of T's and A's in it. And in opening it up, you now form an open promoter complex. There's an interesting fact here between the closed promoter complex and the open promoter complex. Can anyone guess which complex the RNA polymerase is more strongly bound to? The open promoter complex or the closed promoter complex? Which one do you think is the RNA polymerase more stuck to? The open promoter complex or the closed promoter complex? Raise your hand if you think open promoter complex. Raise your hand if you think closed promoter complex. Raise your hand if you're waiting for the answer. Okay. All right. So I'll describe it this way. The closed promoter complex is formed all the time, even if transcription isn't happening. There are times that RNA polymerase will bind to a promoter, but doesn't actually do anything. For whatever reason, we don't know every reason why it doesn't decide to transcribe. But what this means is that RNA polymerase can bind to the closed promoter complex and sometimes just leave. So it does not bind very strongly to the closed promoter complex. However, when DNA actually opens up, everything is now committed to performing transcription. RNA polymerase is now stuck to the DNA template in an open promoter complex because it is committed to transcription. It's going to actually transcribe the gene now. All right? Closed promoter complex, nothing technically has started yet. So it binds very loosely to the closed promoter complex. All right. Well, after opening up the DNA, the first couple bases come in. So this is all initiation. All right, this is all initiation. Probably should have started with that. This is all initiation stuff. Okay. First couple bases are brought in, and I think you have to actually memorize. Let me go to that slide real quick because I never remember it. Exactly which bases. Let's see. All right. Initiation. All right. So the first couple bases that are brought in are A's and G's, which are, anyone remember, are those your purines or pyrimidines? Those are your purines, all right? I think Dervin's came up with a neat trick to memorize your purines, pure as gold, A's and G's, okay? So it turns out in transcription, the first couple bases that are brought in are actually your purines, A's and G's, just a few of them. And then the rest of transcription is whatever base needs to bind to. So it turns out the very start of transcription overly favors sites that will bind to A's and G's for whatever reason that we don't have to know. All right, but memorize these facts about what bases go to the initiation site and which ones go to the elongation site. Let me see if there's anything else. I'll get to these things later. Oh yeah. Remember in DNA replication, the way it works is that you always add nucleotides on the 3' end of a nucleotide chain. That's because the 3' end has a free hydroxyl group on it. Same reasoning here. If you want to add new mRNA nucleotides, you want to add new RNA nucleotides, there needs to be a 3' OH that it can bind to. So that is mentioned here. Let's see anything else. All right, so let's go back to this picture now. So yeah, a purine is brought in at the initiation site, which is like the first site that is being transcribed. After that, it actually transcribes a couple more bases. I think 6 to 10 is, well, this one says 4, the other slide says a couple more. You don't have to memorize the number, just a couple more bases are transcribed. But then it runs into a problem. After transcribing these things, the sigma subunit is still on RNA polymerase. RNA polymerase is stuck to the DNA template. Despite DNA is now open, and despite that it has begun transcription, the sigma subunit is a parking brake. It's keeping the RNA polymerase stuck in one spot. So the very last step in initiation, by definition, is the sigma subunit leaves. Because it's achieved its job. It brought RNA polymerase in, it allowed it to stick to the template, and now we don't need it anymore. So when sigma subunit leaves, the RNA polymerase is now free to move down the template and transcribe the rest of it. So the end of initiation, the end of the initiation step in transcription is that the sigma subunit leaves, so you get rid of the parking brake, RNA polymerase transcribes the rest of this. Okay? All right, so some vocab terms to point out. I will move to that. Let's see, is it? Oh, there should be a slide that describes hollow enzyme, and I can't find it. Is it this one? No. All right, well, they don't define it here. They don't define it, but let me describe what hollow enzyme means. RNA polymerase, a hollow enzyme is all of your subunits put together. RNA polymerase by itself is everything except for the sigma subunit. The core polymerase, the core RNA polymerase, is everything but the sigma subunit. And then when the core polymerase is combined with the sigma subunit, you use this vocab term, hollow enzyme or holozyme, to describe it. Okay? You need the RNA polymerase holozyme to begin transcription, right? We saw what sigma was supposed to be used for. But you only need the core polymerase to actually continue the rest of transcription. You don't need the sigma subunit after you've started everything. All right? Little subtle differences. I know it's annoying, but you have to focus on these things for certain exam questions. All right. Are we getting to elongation yet? I think so. Okay. Oh yeah, some other facts that may show up on the exam. So there's some antibiotics that we have designed that specifically work really well because they target prokaryotic RNA polymerase. Prokaryotic RNA polymerase looks different from eukaryotic RNA polymerase. So by designing antibiotics that specifically attack prokaryotic RNA polymerase, we can kill bacteria that are responsible for some type of infection. These are the names in this example here. And they both bind to certain sites on the RNA polymerase. And their main job is, well, when they bind to RNA polymerase no longer is able, the prokaryotic one, is no longer able to bind to a promoter region and transcribe a gene. So that's what the purpose of these named antibiotics are, so make sure you know their names and what they do. Okay. Oh yeah, and I don't remember if he's given you any other antibiotics names. I think he talked about penicillin a long time ago. Do you guys remember what penicillins, how it kills bacteria? What did it inhibit? Does peptidoglycan sound familiar, right? Penicillin's job is to inhibit peptidoglycan synthesis, which is necessary for proper bacteria, cell membranes, and cell walls, right? These things are also antibiotics, but they do something completely different. So be careful of that. Don't just memorize, oh, these antibiotics kill bacteria. You need to know that these kill bacteria by specifically blocking RNA polymerase. Therefore, you disrupt the central dogma process in prokaryotes using these drugs. All right. The next step, which I think is the easiest step in transcription, is elongation. So why is this the easiest step? Because all that happens is elongation is the enzyme reads the template and brings in a new mRNA base to extend the mRNA that it is synthesizing, or any RNA, I should say. This happens over and over again. You can memorize numbers if you want. Those are not usually asked about. Instead, the following things are asked about in terms of chain elongation, one of which is what this diagram is telling you. So let me zoom in. So here's RNA polymerase. Notice DNA is opened up and it is currently reading the template strand to transcribe it. Remember in DNA replication, though, that there are some consequences with opening up DNA. It turns out that ahead of the transcriptional bubble, which is what this region is called, ahead of it, you create positive supercoiling, which needs to be fixed, otherwise you're going to damage DNA. What particular topoisomerase do we know his main job is to fix positive supercoiling? Gyrase. So, one thing that is often asked about is what enzymes are involved in supercoiling during transcription? Well, gyrase is ahead of the transcriptional bubble, constantly fixing these positive supercoils. And back here, they give a more general answer. You have negative supercoiling in the back that is usually fixed by general topoisomerases that fix the negative supercoiling in the back of the transcriptional bubble. All right. So elongation, again, is very simple. You just extend the mRNA strand by continuing to read the template. You have gyrases at the front to fix positive supercoiling. You have topoisomerase, general topoisomerases in the back to fix negative supercoiling. And let me see if there's anything else important. Trying to recall. Oh yeah, mistakes. So, remember, more compare and contrast stuff, more comparing and contrasting stuff. Remember that DNA polymerase had that activity called 3' to 5' exonuclease, right? Anyone remember what that does in DNA polymerase? 3' exonuclease? What is that? Is that your proofreader or is it the editor? Anyone remember? Is the editor. A 3' exonuclease in DNA polymerases. The 3' exonuclease's job is to edit mistakes. So DNA polymerases, therefore, are very good at replicating DNA consistently because it has a bunch of backup mechanisms to fix mistakes. RNA polymerase has none of those things. RNA polymerase does not have any editing mechanism, does not have any proofreading mechanism. RNA polymerase is just going to transcribe mRNA and hope it gets it right. All right? That's okay because mRNA is not a permanent structure. mRNA is a temporary thing that is made. And even if the mRNA has a mistake in it, it's not going to lead into any crazy mutations because the DNA is still fine. All right? So RNA polymerase, evolution has determined, does not need any repair enzymatic activities or editing enzymatic activities because even if it makes a mistake, later on it'll just transcribe the gene again and probably get it right the second time. Okay? So that's another compare-contrast thing between transcription and DNA replication. RNA polymerase can't repair mistakes. Oh yeah, what about the cleno fragment? Can the large cleno fragment repair mistakes? Yes or no? Yes? Why? What activity does it have that you now can conclude that it can repair mistakes? The 3' exonuclease, right? So more little things that you will see as either answer choices or questions on the exam. You need to think about it in a deeper sense rather than just memorize. You've got to compare and contrast these things. All right. Oh yeah, this is just illustrating the supercoiling fixing and all that. All right. Last bit of transcription. So initiation, we got RNA polymerase binding to promoters, synthesizing a couple nucleotides, and the sigma subunit leaving. Elongation, you extend your mRNA strand, you fix some supercoiling, and you hope you don't make any mistakes. Chain termination in prokaryotes is rather complicated because there's two ways that transcription can be terminated in prokaryotes. This is only a prokaryote thing. All right, this stuff here you will not see in eukaryotic transcriptional termination. This is only a prokaryote thing. And the way they name it is row-dependent termination or row-independent termination. And row-independent termination, you'll see written here as it depends on specific sequences. I'll explain what that means later. So the two ways prokaryotes finish transcription is row-dependent and row-independent. Let's look at a picture for row-dependent. This slide here, these steps, is very important for the exam. You may be asked about any component on this slide here with the steps of row-dependent termination. I will only go over the most important concepts, but there's some specific names of things on here that you might need to memorize. But here's how it works. Row-dependent termination is named that way because transcription ends due to a protein called Rho, RHO. All right? How this protein ends transcription is that it actually was made from the very front of this mRNA. Remember in prokaryotes that transcription and translation can happen simultaneously because there's no nucleus. Prokaryotes can do transcription and translation simultaneously. So it turns out that every time transcription starts, a Rho protein is actually translated while transcription is going on. When this Rho protein is made, it binds to the mRNA and then begins moving up the mRNA. So notice it is here, and notice it is moving up down the 3' end to catch up to the transcriptional bubble. When Rho catches up to the transcriptional bubble, it uses its helicase activity to unwind everything from each other. All right, so that's all Rho is. Rho is a special protein to end transcription by using its helicase activity. So that is Rho dependent termination. Again, you can end transcription because you unwind everything from each other. How can you continue on afterwards? So that is the Rho protein's job. Very common confusion part here is, do you guys remember something called the Tusk protein in prokaryotic DNA replication? Does anyone remember what the activity that the Tusk protein had in prokaryotic DNA replication? Contrahelicase. A very common exam mistake is people get the Tusk protein mixed up with the Rho protein. This is why you write your notes on separate sheets of paper and you hold them next to each other and you compare them to each other, because there's a lot of similar sounding words. All right? I'll go over this in more detail next time. We'll go over Rho independent termination next time. We'll have to end for now. Whoever has a sign-in sheet, please bring it up here.